So, for termination, there are two types of mechanisms in prokaryotes. Either the termination occurs in a row-dependent phenomena, or it becomes in a row-independent manner. Now you must be wondering, what is a row? Rho is an additional cofactor, pretty much like sigma, which we faced in the initiation. It's a protein which is a hexamer. इसके 6 subunits हैं, and it acts as an ATPase as well as a helicase. So you can see in this particular diagram कि किस तरीके से ये जाकर बाइंड करती है एक RNA, mRNA जो प्रोड्यूस हो रहा होता है उसको. In this particular diagram, what you are watching is a row dependent termination. Row factor ultimately slide करता है and moves along 5' to 3' direction, and वो ultimately जाकर उस complex को dislodge कर देता है जिसके अंदर transcription हो रही होती है. But it is not that easy. It's an oversimplification of a very complex phenomena. And ultimately RNA polymerase भी dislodge हो जाता है और row protein भी dislodge हो जाती है. But when we are talking about row independent termination, this particular termination is also known as intrinsic termination. And this intrinsic termination is determined by specific termination sites. So what are the characteristics of these termination sites? These termination sites have inverted repeats, typically GC rich regions, जिनकी वजह से एक स्टेबल स्टेम लूप स्ट्रक्चर बनता है. इनके दरमियान में ताकि इन्वर्टेड रिपीट आए, देयर इज अ नॉन रिपीटिंग सेगमेंट दैट पंक्चुएट्स द इन्वर्टेड रिपीट्स, एंड a run of 6 to 8 adenines is found in the DN template, which will ultimately code for uracils, ताकि आपको क्लियर हो जाए कि बेसिकली mRNA एंड होने लगा है. Let's look at this image. So there is our first inverted repeat, and then we have a second inverted repeat, and in between, in this gray box, we have a non-GC rich region which is not complementary to these two inverted repeats. Now ultimately this inverted repeats makes a bond with each other, and ultimately it becomes physically impossible for the polymerase to not get affected by this particular stem loop structure. So this stem loop disruption, basically it causes a conformational change in RNA polymerase, leading to the pause of RNA polymerase moving. Then the competition of RNA-RNA hybrid and the DNA-DNA hybrid reduces the DNA-RNA hybrid stability, क्योंकि RNA has basically made a bond with its own self, and that causes the transcription complex, that causes the dissociation of transcription complex. And among all the base pairing, the most unstable one is that of uracil and adenine, because adenine always favors thymine. So what you are watching here is that ultimately stem loop structure disrupts and terminated. It can be better seen in an animated video. Let me show you that.
Transcription is initiated just beyond the yellow promoter region and is terminated at the green terminator site. The sequence of the promoter at positions 35 and 10 nucleotides upstream from the transcription start site are critical to initiating transcription. In order to initiate transcription in bacteria, core RNA polymerase and sigma factor form a holoenzyme. Sigma factor recognizes the sequence of DNA at the promoter region. This holoenzyme then binds to the promoter region of the DNA molecule, forming the closed complex between RNA polymerase and the DNA molecule. By breaking hydrogen bonds between bases in the double helix, the closed complex is converted into the open complex. The formation of the RNA transcript from nucleotide triphosphates then takes place as RNA polymerase is forming the new transcript, starting at the 5' end of the transcript. New nucleotides are added onto the free 3' end. The initiation of transcription is completed when sigma factor is released. As elongation of the transcript then continues, the RNA polymerase moves down the DNA molecule and the next sequence in the chain is opened up. As RNA polymerase moves down the DNA strand, the DNA molecule entering RNA polymerase unwinds, forming the open complex, while the DNA exiting RNA polymerase rewinds to form double helix. Ribonucleotide triphosphates are added to the chain, and the growing RNA transcript continues to elongate as new DNA sequence enters the open complex region. The new RNA transcript initially forms an RNA-DNA hybrid complementary to the template strand, corresponding to the sequence of the coding DNA strand. In this way, the DNA double helix is opened, transcribed, and reclosed with minimal stress on the DNA molecule. One mechanism of bacterial transcriptional termination involves the binding of the Rho protein to the RNA. It is a helicase that moves in the 5' to 3' direction toward the RNA polymerase. Between the Rho recognition site and the terminator site, a stem loop structure is formed by the sequence in this region. This stem loop structure halts the movement of RNA polymerase, and the Rho protein then moves to the RNA polymerase complex, causing the complex to dissociate and terminating transcription, resulting in the full-length mRNA transcribed from the gene.
So, in this video you saw that how transcription happens, but there were a few inaccuracies. For example, sigma factor doesn't bind to RNA polymerase beforehand. It's a very dynamic process. On one hand, sigma factor is recognizing a promoter and simultaneously it binds to RNA polymerase. And in the end also, they combined the both systems in termination, that how Rho and stem loop structure works. But you have to understand that these are very dynamic systems, and they, in these particular videos, they are explained in a way so that you can understand. If we will depict them in a very dynamic environment, then you won't be able to understand them.